So our next speaker is, I'm extremely happy that he accepted our invitation. Someone who is very important and will help in this, in our movement towards changing health policy, and hopefully will help to fund important research that will be necessary. Dr. Asad Umar is from the National Institutes of Health. He is the chief of the Division of Cancer Prevention in the National Cancer Institute. Fortunately, he has a particular interest in GI malignancies and has funded multiple large research grants related to GI malignancies. And so my hope today is that he gives us great news and guidance on how to improve our funding and what we can do to increase funding, NCI and NIH funding for the study of gastric cancer. So welcome.
Thank you very much for this kind invitation by the organizers and kind words for my introduction as well. Hopefully, I will be able to fulfill some of those promises or things that you were expecting, but things don't work that fast at the federal level, as you are aware of that. But I will give you a picture of at least what our thinking is, what we are doing. We definitely are very much interested in the gastric cancer prevention and overall thinking of just cancer prevention as you heard that my interest is in cancer prevention and especially GI cancers, so gastrointestinal cancer prevention research group, what I lead. We think a lot about gastric cancer and have few clinical trials that are ongoing. This is one of them you will hear tomorrow more detail. The other one I'll mention. But there's not a whole lot going on, but at least the overall picture where we are and where we might be going in future, hopefully in near future. So the way I think about cancer prevention is for, if you look at it on a time scale, for interventions during carcinogenesis, you can intervene at different times. So for treatment, you have a survival with cancer, and the goal for cancer prevention is actually intervene before that cancer is invasive, and earlier and earlier we can identify, as other speakers have talked about, we can actually have a survival without cancer. And these precancer, we can reverse the precancers or remove surgically these precancerous lesions. So that's the hope. And we have seen different versions of this slide, the timeline and progression of gastric cancer and pre-malignant stages for gastric cancer, from normal epithelia to gastritis, atrophic gastritis, intestinal metaplasia, dysplasia, and adenocarcinoma. A number of risk factors, H. pylori, we just heard about that, in additional component that might play at same point or synergistically. Some of these things like high salt consumption or food that's preserved with salt, or smoking, antioxidants or oxidants might be blocking, antioxidant might be blocking that pathway, and how we can intervene in all these different molecular changes that are going on during this carcinogenesis pathway. The estimated number of new cases worldwide, as were also talked about by several speakers today, overall stomach cancer is pretty high. Overall, it's like the fifth cancer that's on the list of total overall in the global CAN numbers. It's not the same burden throughout the world, as you've seen different versions of this global chart as well, where some of the areas like the Asian countries, as we just heard, are the highest incidence, Japan and Korea being the highest one, again highlighted in this. is the Japan, Korea, South America, and Eastern Europe are the highest. Adjusted rates worldwide, if we look aggregate, are 15.62 per 100,000, and for Latin America is a variable, as well as other countries is also variable. But we also heard that the United States, the incidence has been decreasing, and the disease has migrated proximally. Maybe this is a change in the gene and environment, or what gastroesophageal lesions are more frequent than the antral lesions more recently. So to summarize the risk factors for gastric cancer, H. pylori, Epstein-Barr virus was recently, relatively recently, was identified through the TCGA effort to be also associated with, although smaller number, but still significant number of gastric cancers. Smoking, the strong evidence for alcohol, especially with the ALDH2 polymorphism, food preserved with salt, obesity, and the connection of obesity probably is with the inflammation and IL-6, TNF-alpha, and the C-reactive protein, as well as insulin. There is some evidence for processed meat and grilled and charred meat, but it's not that strong evidence for those to be associated with gastric cancer risk factors. So for primary prevention, the way we think about for prevention is reducing exposures to these risk factors, or by increasing the resistance to these risk factors. So some of these, the efforts at NCI and as well as other agencies, like the smoking cessation, and also with simple hygiene, and how that has affected the overall gastric cancer as well as other cancers in that regard. For secondary prevention, early screening and early detection and screening is important. It seems like from what Chen was saying, that maybe we are there for screening, and maybe there is some work is still needed. We definitely need a lot more work for early detection, and to be able to really actually attack the disease, and probably is a really very good case for having a screening and early detection of gastric cancer, because it seems like this is preventable. For tertiary prevention, referring to treatment of risk factors, for example, H. pylori or EBV treatment, or prophylaxis with possibly vaccines or with antibiotics, or precancers to reverse the course of disease. So for primary prevention of gastric cancer, H. pylori infection eradication, as you heard, I also have highlighted meta-analysis of 7 randomized clinical trials have shown that high gastric cancer risk areas suggest the eradication of H. pylori may reduce gastric cancer risk to a relative risk of 0.65. In Shandong, China, an extension of an intervention study for 14 plus years, saw a reduction in incidence of gastric cancer by 39%. Mortality was reduced in the study 1.5% versus 2.1, and hazard risk was 2.67 in that case. Smoking cessation has also been associated. The risk of cancer is decreased with smoking cessation. Diet with excessive salt increases the risk of gastric cancer by at least twofold. And some of the animal models have suggestion that actually there is a synergistic effect of H. pylori infection and gastric cancer with salt, high salt food diet. Increased intake of vegetables and salt has been estimated to reduce risk factor by 30 to 40% respectively. So there is no, but overall there are no conclusive data on dietary changes that much as far as H. pylori and smoking cessation is concerned. So there seems to be, there's a number of things that can be done even now without going into additional chemopreventive strategies. But for H. pylori eradication, for example, on this chart is showing the meta-analysis of these clinical trials. You can see there's overwhelming evidence favoring the treatment eradication with antibiotics leading to benefit. Other interventions like anti-inflammatory drugs intake in gastric cancer, aspirin, for example, number of reports. This is, again, just a compilation of different studies that are showing a variable data for frequency seems to be higher the frequency of intake of NSAIDs or aspirin is, the relative risk is lower with the higher frequency times per month of aspirin intake. And same is true for COX-2 inhibitors. So not just the aspirin, so the non-specific cyclooxygenase inhibitors, but COX-2 specific inhibitors also showing this reduced relative risk reduction in case of gastric cancer, as well as other NSAIDs. So it seems to be anti-inflammatory are one of the areas, but that it has to be balanced with aspirins being also an irritant, especially with the H. pylori infection at the same time. That is a consideration for doing a clinical trial in that setting for gastric cancer. So we are, one of this trial that, as I mentioned, is looking at a drug called Meriva. It's actually a supplement, which is curcumin, which has similar properties in terms of inflammation, anti-inflammatory. So it's anti-inflammatory. It's some effect on NF-kappa B as well as a lot of the inflammatory cytokines. So Meriva is a nanoparticle or liposome encapsulated curcumin. Problem with curcumin is we have done a lot of trials with curcumin looking at a number of GI tract where we don't really even need absorption, that the curcumin doesn't really get absorbed. So curcumin but can be topically applied to GI tract, at least when you take orally. Curcumin is the part that is actually is curry in a lot of the foods and Indian cuisines and other cuisines too. But so there is a lot of data on that in animal studies as well as some clinical trials. We have not been able to find really an active component in curcumin, predominantly because of not being absorbed. However, Meriva, on the other hand, being a liposomal formulation, does get absorbed at a higher, much higher rate. It's 27-fold higher compared to curcumin alone, so blood levels can be seen. So we have a hope that this will actually work in the population. The study population is a small study where we're just really looking at the IL-1 beta changes, cytokine level. That's a biomarker study. It's a phase 2 trial. It's in the individuals 21 and older. It's being done in Honduras and Puerto Rico. The PI is Doug Morgan, and Keith is part of this study too. If I don't remember it correctly, he can correct it. So the Meriva is 500 milligram BID versus placebo taken alone or in individuals that have a surgical history or laboratory studies or have a stool antigen test positive for H. pylori, as well as some EGD and biopsies will be taken from this study. And after the intervention, the IL-1 beta level is the measure for actually Meriva or curcumin going into the bloodstream and having absorption and enhanced plasma concentration of these curcuminide profiles are our final endpoint of this study. Other than that, I think Keith is going to talk about polyamines and DFMO, and I'm not going to put any of that. That's another study that we are supporting, and we're very excited about the data that Keith Wilson is going to talk tomorrow about that. So stay tuned for that. So other things that we are working on, and some of the things I've listed here is just H. pylori eradication, another study that was done by SWOG. Also showed, again, a reduction in the the cancer incidence. However, there was a high incidence of infection in those individuals. DFMO, that I just mentioned, Doug Morgan and Keith Wilson are doing that in H. pylori positive gastric precancers. And Mariva, and also we are working on an EBV vaccine at the NCI with a company called Inovio that has a very nice platform to quickly move DNA-based vaccines into clinical trials. It's actually the same company that's developing a vaccine for coronavirus right now, one of those companies. We are also thinking about using one of the vaccines that we have developed and is ready to go in clinical trials is based on frameshift peptides for Lynch syndromes, because it's a reasonable number of cases have microsatellite instability for gastric cancer. And this microsatellite instability is all that's needed. Microsatellite instability leads to frame shifts in the normal proteins, creating new antigens. So those neoantigens have been identified due to the mismatch repair defect, and we have actually candidate peptides that we are working with a number of vendors to have a CGMP vaccine ready in the next year or so that will be primarily for Lynch, but it could be also used in a MSI positive, along with once the MSI positive, also with the checkpoint blockade as well. With that, I'll stop here and thank you again for your attention.